I hope you're still willing to listen to me. So now we will discuss another revolution. It's the day of revolution today. And we are discussing the emergence of another revolutionary mechanism by which antigen receptors can somatically diversify. And this process is called VDJ recombination, and you probably will have heard about this, but I will explain to you what it really means in simple terms and to explain also the name of it. This is now a more sophisticated type of review of a picture that I drew on the blackboard. We discussed a little bit these jawless vertebrates, and there is evidence now that hagfish also have these three types of VLR receptors that lamprey have, indicating that they both are originating from the same common ancestor. And at some point after the jawless vertebrates split off from the jawed vertebrates, people think that a genetic transposition event occurred, a transposon, the identity of which has now been nailed down to be a so-called transip transposon, invaded the genome of an early jawed vertebrate and brought in what we nowadays call RAG recombinases. Because as you know, transposons, they have, among many things, also enzymes that allow them to splice themselves into the genome and also remove themselves from the genome to jump across the genome or between genomes. And the very peculiar genomic organization of recombination activating genes, RAG genes, of two genes next to each other in a particular genomic orientations indicated early on that they might have originated from transposons. And the second indication of why that might have happened is that the recombination of antigen receptors in jawed vertebrates, that is immunoglobulins and T-cell receptors, were very soon shown, or soon after the discovery shown, to depend on so-called recombination signal sequences that looked very much like sequences one knew from other studies to be flanking mobile genetic elements, such as transposons. So there was this early speculation, and we simply have to take this as speculation for the moment, but I think there is good evidence for this, that this must have occurred by lateral gene transfer, although I should say in parentheses that RAG-like genes have also been found in invertebrates. Some people argue this means that there is shared ancestry, and jawless vertebrates and many other species in between have lost these genes, or else you could say that these transposons invaded different genomes at different points of time independently. But be this as it may, these recombinases do something really amazing. Apparently, when one follows the evolutionary trajectory, the first transposon insertion event occurred in what we call a primordial antigen receptor gene, so IGTCR gene. In the beginning, of course, they probably looked alike, and it probably was a single gene that this transposon inserted into. So it inserts into a gene that has a proto-immunoglobulin superfamily domain, a variable domain. I explained to you this is characterized by a certain structure and length of amino acids with this single disulfide bond. A particular sequence of amino acids that we now term joining sequence for a number of reasons, a transmembrane and a cytoplasmic domain. So the assumption is that this transposon invaded this gene, that is a contiguous gene, in between this variable portion and this joining portion. So this is meant to be the transposon, and this is these direct repeats that flank the insertion site of mobile genetic elements. Now, if you see this, and microbiologists use this, this is what is called insertional mutagenesis. So the gene is non-functional. So you insert a piece of genetic information that is unrelated to the original gene. You do this, gene is dead. Okay? So that's very well, and that might have happened numerous times. Most of the time, it simply led to inactivation of genes, and if there is a mutation, a non-functional gene, it's lost by Darwinian selection. But in this case, maybe something else happened. The function was restored by excision of the transposon. So, we insert, we leave the gene, this transposon in the gene, and now we are removing it. And this is also known from microbiology or genetics. If you remove a mobile genetic element, most of the time, depending on what kind of element it is, it leaves a scar. So you can tell where this transposon was and has left the genome. And the scar is depicted here in this little colored piece. So what that suggests to you now when you look at this, you make a non-functional gene, you restore activity, but you generate something that is not like the original version. It's something different. And this difference can have important functional consequences because it might change the coding capacity of this gene. Now, I explained to you yesterday already a double-strand break that occurs in the cell is not much liked, and it's immediately repaired by a mechanism that is called non-homologous end joining. And this non-homologous end joining is like an emergency. It's like basically, you probably know this television series, Emergency Room. There's not much differential diagnosis when somebody is brought in. It needs to be acted upon quickly, no matter what the underlying cause is, get the guy going again, okay? So and this is the same here. We close the gap, we repair it at the cost of introducing mutations. So when this transposon leaves its insertion site, it creates a double-strand break, it leaves a scar perhaps, but it certainly leaves the remnants of this emergency sealing event, this non-homologous end joining. And this repair process, although it's very efficient and very quick, it creates basically random mutations. But these random mutations are now very localized because they are not, like in somatic hypermutation, scattered across the entire gene or an entire genomic region. They are really very localized because where this break was, there will be the mutation. Now, in the context of an antigen receptor, and if the antigen receptor happens to have a structure where the site of excision, that is this site of randomness, is involved in binding antigen, this has a great advantage because you can now make a mutated or a varied form of this antigen binding site. And if you do this in a somatic cell, this is your germline configuration. In lymphocyte 1, you make this change. In lymphocyte 2, you make this change. Amazingly efficient procedure. One gene, one process, and each outcome is unique. So in each lymphocyte where this happens, you make a different type of receptor. All the proviso that we discussed before with the VLRs, of course, not always is the sequence different or the protein is really different. Never mind, the process and the principle is there. We can now somatically diversify this locus by removing an inserted intervening sequence and sealing the break with random elements. And this is now the comparison. We've discussed LRRs, gene conversion. You bring in these elements in unspecified number in principle at least, and we have some sort of junctional diversity, but it's mainly combinatorial, but we have enormous diversity in these mature genes. And now we have the same story here. Now we have the V element, the J element, we fuse them together, we have this variable region here. It's called complementary determining region number 3. This is for historical reasons. This is one of the main regions that contact antigen. This is, of course, derived from structures of these molecules, immunoglobulins or T cell receptors, and in conjunction with their particular antigen. But the important point here again is we have combinatorial diversity. If we have random mutagenesis associated with this combinatorial diversity, because you can have many V's and many J's, so you can make many VJ combinations in each time and in each process, in each lymphocyte, a different pink region here, as it were. So you can, by making this break fusion process, many different isoforms or isotypes of your receptor. Once again, so like in lampreys, you fuse these things and you bring them together. And there's two different types, just out of interest. This is the primitive form, duplicated VJC. We had that before. So we had a constant region that is the transmembrane and the cytoplasmic domain. We have this J and the V. So if we can either use this en bloc duplication to make many of these, or we can duplicate internally V, 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 J, 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 and normally one C, but we can also duplicate the C's. And then we have what is called a translocon configuration, because we are going across a locus. If we use this one, for example, and use this one, all the rest in between is gone. This is, of course, one other possibility. So, or we have a cluster. Each cluster is entirely independent and can rearrange. So that is the mechanism of diversity, and because this process involves V-like region, variable regions, and joining regions, the process that is executed by these RAC recombinases is called V-DJ recombination. The D comes from a further elaboration of these loci. There are additional diversity elements that operate by the same principle, situated between the V's and the J's. So that makes then the V-D-J recombination. You can easily forget all the details. You just have to remember VLRs and immunoglobulins and T cell receptors somatically rearrange, and the outcome is not always predicted by the germline coding capacity. That's the key thing. We have an enormous diversity, but the diversity is not predictable. So we skip this here, and you will not be surprised now that we come to this final comparison. We discussed the jawless vertebrates and all these hypothetical ancestors. Now we compare the jawed vertebrates, where we have a lot of information, and the lampreys. Two different T cell types, alpha, beta, gamma, delta. We'll come to this a bit later, but it's not really important. Safe to say that they might have, or almost certainly have, different functions. In bony fish, we even have genetic evidence for two different types of B cells. Perhaps that is also true for lamprey, but we don't have very good evidence, genetic evidence at least, for two different types of B cells in mammals, for example, although there are some indications. So it's basically a nicely symmetric differentiation tree, 2T, 2B, or maybe one or the other lost, but it's amazingly similar in all vertebrates. And I re-emphasize, cell lineages are similar, somatic diversification is similar, unpredictable outcome of somatic diversification is similar. What is not similar is the molecules. And because the molecules are not similar and the mechanisms are not directly comparable, this must mean, as I said many times before now, that the ancestor of these two groups of animals either had an entirely different somatic diversification mechanism or none at all. And that is a very important conclusion. And that was only possible, that conclusion, because the Cooper laboratory discovered this alternative form of antigen receptors in jawless vertebrates. So we now know that the common ancestor had neither of these, or you could say maybe it had both. yet it was then different from the other two branches, or it had none, or it might have had one, but then it's a bit unlikely this was reinvented. Normally when things prove useful, they like to stay, but this is now up to speculation. You can make all possibilities. The beauty of this is you will never be able to verify this, because this animal no longer exists. And you can put that down in a bit more detail, and you will see then that the common vertebrate ancestor had a proto-IGT-CR, so the one that was invaded by the RAG recombinase, or this particular transposon, already exists or still exists in the cyclostomes, so the jawless vertebrates. The CDAs, the cytidine deaminases that are so important for the somatic diversification in this group of animals, already exists or still exists in our group of animals, and is also used, I alluded to this briefly, for somatic diversification, hypermutation in the B cell lineage. This is the ancestor. This glycoprotein here is the ancestor of the VLRs, and this still exists in our genome. The only discontinuity is the introduction by lateral gene transfer of these RAG recombinases. So we have a fairly good picture now, and I think this is a very satisfying conclusion that emerged within the last 50 years or so, of how immune systems and different antigen receptor types evolved, and how they are functionally similar and different at the same time in building the very efficient immune system of these two types of vertebrates. I think I'll skip over this here in the interest of time. Now, many of you might have heard about monoallelic gene expression. And I'll give you one potential reason for this. I talked about economy in the immune system, or economy in living vertebrates, in any animal basically. You want to save energy. Energy is very costly, so you want to do and go about with least amount of ATP. And anything you do in biosynthetic pathways costs energy, so you want to save energy. So when you then think about the development of these lymphocytes that we had in this very nice cartoon, you start from an incomplete and basically useless antigen receptor locus in the germline, either the incomplete VLR or the incomplete immunoglobulin TCR locus. You can't do anything with this. You have to assemble it to make it work. That is the process of somatic diversification at the same time, but it also brings in functional receptors. Now, we also talked about the danger of self-reactivity, and that is also not good, because you don't want to have receptors that attack your own tissue. And that is perhaps inevitable when you make these random mistakes during the fusion process. So you have to find ways of building a quality control. I talked about the car factories and the car manufacturers. They have to build in quality control, check the car before they sell it to the customer. But sometimes that goes wrong, as you all know. So then some car manufacturer places ads in the newspaper or on television, bring back your cars because the brakes don't work. You might not have realized, but we have to fix this. That might be possible because we can listen to television, but that is not possible in the immune system. Once the cells are faulty and out in the body, we cannot retrieve them. So we have to build a fail-safe quality control system. And at the same time, we have to be efficient. So we cannot say we are making 100 lymphocytes and only one of them is useful, because building 99 lymphocytes for nothing costs a lot of energy and doesn't bring anything, no advantage. But of course, as always, there has to be a compromise. And the compromise is we want self-tolerant receptors and self-tolerant receptors equipping lymphocytes, and at the same time we want to avoid waste of self-reactive, autoreactive lymphocytes. Now, what is an efficient strategy to do this? The solution to this is monoallelic expression. Each lymphocyte expresses only one receptor, and you will very quickly see why that is the case. It's a calculation you can do on the back of an envelope, very simply, and you have a very obvious and and very convincing explanation for this particular situation. Because not only in the lamprey, also in the mammals, whenever one studies lymphocytes to a first approximation and in general, each lymphocyte has a single receptor. And intuitively, that might not make sense, because you might say, okay, I have too few lymphocytes anyway. I have so many different receptors. Why do I not use these receptors, many of them, on one lymphocyte? Because then one lymphocyte is polyreactive and can react against A, B, C, and D, and the other one against C, D, E, F. And that covers a much greater spectrum of antigens. That might be quite advantageous. But then, of course, the problem comes when you do this. You might have one receptor on that cell among the 10 that you are expressing that is self-reactive. So the 9 good ones are overpowered by the one bad guy, because that is self-reactive, activates the lymphocyte irrespective of the good receptors, as it were, the self-tolerant ones, and then you have a problem. So the only solution to this is you express one receptor per cell. So this is the monospecific expression of antigen receptors. This is what people think now the key sign of an advanced, some call it adaptive immune system. All the other criteria we discussed yesterday are not useful. The only useful definition of an advanced immune system is monospecific expression of antigen receptors.
But maybe there is another explanation, that if lymphocyte activates, then it dies, and then 9 receptors are useless.
Say this again, one.
But if lymphocyte is activated, then it consumes and dies, and then the 9 other receptors are useless. Can it be so?
Okay, so the argument here is that if a lymphocyte is activated, it dies. Well, that's not quite true, because there is this phenomenon of immunological memory. And that means a lymphocyte recognizes its antigen, proliferates, maybe if it's a cytotoxic T cell, for example, then kills the target cell. But some of these activated cells then change phenotype and become long-lived so-called memory cells. And next time you are attacked by the same antigen, as it were, then they can proliferate, or they can react much faster, or the immune system as such can react much faster. So that's the memory effect. That's the vaccination, the success of vaccination. Yeah? So I don't think you can always, of course there's a possibility if you activate a lymphocyte it might die, and there are examples for this, but in general the lymphocyte is activated, proliferates, does its job, and then moves into a different compartment, as it were, to prepare for future challenge. Now, if we assume now that the outcome of antigen receptors at the cell surface being self-reactive is not desired, because we want to avoid autoimmunity, then you can do the following calculation. So what I've done here is I've depicted the fraction of self-tolerant cells, self-tolerance. So all cells are self-tolerant when the point is here. None of them is self-tolerant when the point is here. Against the number of expressed antigen receptors, in this example from 1, there's only one possibility, or 50. And against the average self-reactivity. So that, of course, has to be an average value. So you do your rearrangement, and then that's what it is. So if you have many options, and there are some examples, I showed you the cluster arrangement, translocon rearrangement, so you can certainly make 2 receptors because you have a biallelic structure. If you have a translocon, if you have a cluster structure like in cartilaginous fishes, you can make 50, 100, or 200 antigen receptors per cell because you can rearrange many, many loci. So this is this example here. And here is 100% self-reactivity. So there is nothing in the repertoire that is self-tolerant. This is towards 0 probability of self-reactivity. Almost all cells are self-tolerant. So you can quickly calculate the fraction, and you can easily see, I mean, that's intuitive, but that's just the numerical solution to this. When you have only one receptor, that is 2 forms expressed, with a certain probability of self-reactivity, the fraction of self-tolerant cells is always maximal. This is always the best outcome.
Excuse me, to be clear, when you say one, you mean it's one gene, but both alleles expressed? So it's actually two different...
Yes, but one gene.
Right.
One gene. So this is one gene, both alleles, biallelic expression, co-dominant expression.
Right.
Okay, so this is the outcome. And you can see, when you, for example, when you have 50 genes, and you have even if you have a probability of 1% of self-reactivity in total of these 50, there's basically nothing in the repertoire. So this is really a wasteful exercise.
Is there any specific reason why DCR alpha locus is an exception? Do you think there is a logical reason for that, or it's just
Now this is detail, we don't discuss the detail. Now when you translate this thinking into the diversity of the repertoire, because that of course matters, you want to have as many receptors as possible for as few lymphocytes as possible. So when you do this here, now this is the diversity, and this is now calculated for 1,000 cells, just to illustrate it. So you have 1,000 cells, you have 2,000 cells here, and it's the same logic. So when you look at the self-reactivity, the number of lymphocytes, so you see here, of course, when you go towards zero of the probability of self-reactivity, the diversity, of course, is maximal. If you go to 50, 50 times 2 is 100, times 1,000 is 10 to the 5. But you see that these curves in this range here, between 20 and 40%, change, and then as of 40%, number one wins.
But I wonder, why don't we have different kinds of lymphocytes? Maybe most of them may express one antigen, and maybe there are very few that can express a lot of antigens.
Yeah, but the same logic applies. This is simply now a summation over a random population. Okay, so it's just a normal distribution. Some make all of them, some make few of them. So it's basically the average that you take. But the outcome will be the same. It's always at a certain probability of self-reactivity, the diversity of the resulting self-tolerant repertoire will be maximal for one co-dominant expressed locus. You can do the calculations tonight and you tell me tomorrow.
Excuse me. Does it mean that probability of cell reactivity to host organism in real situation is more than 14%? So if you take a not selective lymphocyte, it means by this picture that probability of cell reactivity of it would be more than 0.4 in real situation.
That is the key question. Very good question. When you do this for unselected lymphocytes, indeed for immunoglobulins and for T-cell receptors, the fraction of self-reactive lymphocytes is between 30 and 50%. Amazingly high. Of course, that is dependent on the assay system, and you can debate a lot about the value of the assay system, but it's not a very small probability. It's a quite significant probability. And when I go on now and change from co-dominant expression of one locus, that is two different forms, to biallelic, that is co-dominant or monoallelic expression, we do the same logic. Monoallelic expression has the best fraction of self-tolerant cells under all conditions. And these two curves generate, or monoallelic expression generates the highest diversity when it goes above 50%. So because there is such high preponderance of self-reactivity of these unselected receptors when they are being made, the system inevitably is driven towards monoallelic expression, because that is the most efficient way of generating the most diverse repertoire with the least number of lymphocytes. And the least number of lymphocytes means the least expenditure of energy. So it's a very simple logic, and you might agree or not or not agree with it, but I think the conclusion from this is a very important one. Because now monoallelic expression is the rule in all lymphocytes of jawed vertebrates, and monoallelic expression is the rule for lymphocytes in jawless vertebrates. We can hypothesize that lymphocytes in jawless vertebrates per se are also autoreactive. And if that is true, so if my argument is correct, then we would predict that an unselected VLR in jawless vertebrates is also potentially autoreactive. And if that is so, and since lamprey live happy lives, they must also have a quality control system that eliminates self-reactive clones. Okay? So now we have to concern ourselves with the mechanism by which this quality control can happen. And I've used the example of the car factory, and that is indeed how the immune system regulates itself. It couples development to quality control. Nothing else makes sense. You build your car, and before you leave it, before the car leaves the factory, you quality control it. It's no point in sending it out to the individual sales points and then do the quality control there, because if you find a systematic problem, you have to bring them all back again. So you have to couple production to quality control. And now it's a very easy leap in my argument to say that the morphological basis of quality control is what we call a primary lymphoid organ. That is the organ where lymphocytes develop and where they presumably are also quality controlled for self-reactivity. We know that this is true for jawed vertebrates, because we know, for example, it both in the bone marrow and in the thymus, where we understand the mechanism best, T cells are tested for self-reactivity, and if they are self-reactive, they are either functionally eliminated or even destroyed, or in other ways mechanistically silenced. And we come to the mechanisms a bit later. But the end result is that the level of self-reactivity that might remain is tolerable to the organism. There might be some arguments why a certain level of self-reactivity is beneficial, but for our argument today, it's important to conclude self-reactivity is essentially avoided by a quality control mechanism that is coupled to the production of the lymphocytes and the receptors themselves, because it's somatic diversification. Where a germline encoded, we can let Darwinian selection operate. We don't have to worry. We don't have to make any provision for this anywhere in the body. It can be basically any tissue. Think of the myeloid compartment in the hematopoietic system. There is no quality control required because all the receptors, everything they have on the cell surface or inside them is germline encoded. They can develop anywhere. I alluded to this, in the brain, in the lung, or in the bone marrow. It doesn't really matter. But for the lymphocytes, they are potentially dangerous because they can destroy tissue. They can really inflict damage. This is why there has to be a proper quality control. Now our conclusion is monoallelic expression is indicative of self-reactivity, and that this is why we have to conclude that the VLRs are also potentially self-reactive. But there is no evidence. But that sort of line of thinking encouraged us to look for the thymus in lamprey, because we argued if that is really true, then we should expect the presence of a thymus. It took us a long time to find it, but I explained to you in the very beginning that we eventually did. Now, you see that I asked for a little more than I was capable of achieving today, but nonetheless, I will just whet your appetite for tomorrow. Why would we want to study different animal models? I said this yesterday. there are the key models for immunology: humans, the best studied species on Earth in billions of numbers. Mice, of course, because we can manipulate them quite nicely and traditionally very important. Traditional models are also chicken and amphibians to a certain extent. And more people are beginning to be interested in fishes. I explained this, this work in lampreys. I only briefly mentioned sharks, or rather not at all. And I will come then perhaps tomorrow to discuss some really exciting experiments in bony fishes. And the reason why I put up this slide here is just to give you an illustration. We have 100 species of jawless vertebrates, lamprey and hagfish, 1,000 of cartilaginous fishes, 30,000 of bony fishes, 7,000 amphibians, 9,000 reptiles, 10,000 birds, and 5,000 mammals. So amazingly, bony fish are the largest group of these jawed vertebrates. And when you think about them, they live in quite extraordinary different environments. They can live somewhere deep in the sea under extremely high pressure. They can live here in the Neva. They can live in the Baltic Sea, in the Atlantic, God knows where. So they occupy enormously diverse ecological niches. And they are very diverse in morphology. And there is no reason to assume that they're not also very diverse in all other phenotypes, such as the ones that we are interested in, for example, the immune system. So it has some value to study diversity in these individual groups. In mammals, I said to you, we are studying only two, basically, humans and mice. It's rather appalling, actually, but it's 2 out of 5,000. And it would be very difficult to convince anybody that this is a representative example. Okay? If you were to predict election results from asking just 2 people, it probably would be difficult. But it's even worse. We have just 1. Domestic chicken, Gallus gallus. Okay? Well, it's even worse. 1 out of 10,000. Reptiles, basically not studied at all. Okay? Amphibians, 1 or 2 frog species. Bony fishes, and I'm appalled to say, but I have to admit, only a handful that are now under investigation. Cartilaginous fishes, a handful, and here also a handful. So we're doing very poorly. We're not using the richness of diversity in the tree of life to study alternative solutions to the same problem. And we now know that this is very important, because we know that lamprey have solved the problem of somatic diversification and making antigen receptors in quite different ways from the jawed vertebrates. And of course they have a different evolutionary history, although it's connected, but nonetheless there are substantial differences. And you might even argue that some animals might have come up with some extraordinary solutions to particular problems. And I will give you a couple of examples in the days that follow. So it's very important that we appreciate the diversity and at least make some probing into the diversity to see what we can do. And from an experimental point of view, it's also quite useful. And I will give you a couple of examples with the bony fishes, because we can now very nicely genetically manipulate bony fishes. And there are 1 or 2 model organisms that are quite widely used, for example, zebrafish, where we can do precise genetic alterations, we can interfere with development, we can observe these fish under the microscope and can watch them and film them for 24 hours, 48 hours, 72 hours. We can track individual cells over these long time periods and see how they interact with other cells. So we can do a lot that is not possible with our mammalian friends or with our birds. Birds, there are some other advantages, but the fishes offer something that is really unique, and I will give you a couple of examples what we can do. So the greatest species diversity in vertebrate groups is in fish. They occupy these vastly different habitats, and they almost certainly have unique adaptations in their physiology to counter the adverse environments they encounter. I'll end with this to whet your appetite for tomorrow. This is a list that I've taken from one of our own reviews about which genes have been discovered and found to have similar function in the immune system of lower vertebrates, that is fishes, that we know have a function in the immune system in humans based on specific clinical phenotypes. So think of, for example, RAG1. RAG1 is the recombination activation gene. If you have a mutation in RAG1, you cannot assemble your antigen receptors. So it's very easy to predict what might happen. You don't have immunoglobulins, you do not have T-cell receptors. So these patients do very poorly. And when we eliminate RAG functionality in lower vertebrates, we know that we also abolish VDJ recombination. No immunoglobulins, no T-cell receptors. Surprisingly, however, these fish without RAG survive quite happily and live happy lives. They are a bit compromised, but they can do quite well without the lymphocytes that we so strictly depend on. So that already tells you, although the function is the same, the physiological outcome can be quite different. Another one, CXCR4 is a chemokine receptor, a molecule that reacts to small molecules that are secreted, so-called chemokines, and guide interaction between cells. When this receptor is mutated, and we've heard one example yesterday, the CCR5 receptor, when that one is mutated, you abolish infection by HIV. So it is a receptor that is not only used for communication, but also for entry by certain pathogens. But nonetheless, in this particular case, CXCR4, this chemokine receptor, when that is mutated, it leads to increased activity. It's an activating mutation, and patients have a certain phenotype that you can reproduce in lower vertebrates. Another example, IL-7 receptor. IL-7 receptor is a very important mutation in humans. It is a receptor for a cytokine that is very important during the development of lymphocytes. Without IL-7 or IL-7 receptor, lymphocytes develop very poorly. If you have a mutation in that gene that is non-functional, you basically abolish your immune system or immune function. These patients in humans are known as the bubble babies, those that have to be protected from the environment and have to be raised in completely sterile environments. And they have been subjected to various forms of treatment, including gene therapy, where one has tried to insert a functional copy of the IL-7 receptor pathway into the hematopoietic stem cell, and then rebuilding the hematopoietic compartment of these patients with this genetically corrected hematopoietic cells in order to rectify this particular phenotype. So when you do this in fish, for example, you also reproduce the phenotype of T-cell deficiency. You can go through that list and you might not be able to read them, but in each and every case where these molecules that are evolutionarily conserved are associated with particular phenotypes in humans, these phenotypes are mirrored in lower vertebrates. So there is a lot of commonality, and that is encouraging because we get a general feeling for the operation of the system, but then we can be more confident in looking at the anomalies, where there is a difference. Because the difference, of course, when there is a difference between two different species, for example, then it really becomes interesting, because then we can see that there is perhaps the opportunity for alternative solutions. So let me just quickly look at this here, and I think I'll stop here and continue tomorrow. And once again, I hope to see you tomorrow. And tomorrow I'll promise I'll show you some videos just to excite you a little bit. Thanks very much. Questions? Yes, there are questions. Please.
Three questions. My first question is, what do you think is the driving force for the appearance of adaptive immunity? There are some ideas that the driving force is commensal bacteria that lives in the body. What do you think about this?
Yes. A quick answer to this. Yesterday I already discussed that point, and I'm quite convinced that this is one way of explaining the emergence of somatic diversification of these particular types of receptors, because it would have allowed the elaboration of a rather species-rich symbiontic microbiota associated with vertebrate hosts. So
Then what is the difference between lamprey and tunicatus? If they first achieved these elements of adaptive immunity, they both live in water. So when was this border? No adaptive.
yes, when was it crossed, you mean? Well, a simple answer could be perhaps that the genetic capacity that is anyway costly to maintain could be used to elaborate more complex phenotypes. Our body plan is much more complex than the one of tunicates, for example. So you could say, because we managed to build or enhance our metabolic capacity by elaborating a microbiome that's very diverse in function and in species diversity, we could save genetic energy, say, or information to build more complex body plans. And that allowed us to invade new ecological niches and then diversify. Or you could turn the argument around and say, because it was possible, then we had to build and elaborate a symbiotic or more complex symbiotic microbiome. It goes both ways. We don't really know how it was, but I think we can make experiments now. We can, of course, look at impaired diversity in higher vertebrates, even in lamprey perhaps at some day, and then see what the outcome is in terms of microbiome. So if I eliminate, for example, somatic diversification, hypermutation in immunoglobulins, and class switching, I have a very species-restricted microbiome, basically a dysfunctional microbiome in my intestine. That's already known. So it probably is an explanation that perhaps one generated this superorganism with a lot of synthetic capacities by elaborating the diversity in these receptors. One explanation.
You told a very interesting thing that in lamprey there is a transplantation immunity which when these animals can reject homologous skin.
Yes.
So how does it, it cannot be influenced by bacteria. What do you think is the biological sense of transplantation immunity in these animals and in general? Why it was necessary? And what is this, the process to reject the skin? What was this for?
Well, I mean, transplantation I don't think was an idea that occurred in nature, certainly not for vertebrates. We have phenomena that could be discussed in that context, that is allorecognition in colonial tunicates, for example, where we have histocompatibility systems that allow these animals to decide whether they belong to the same genotype or not. But I don't think skin transplantation or organ transplantation was envisaged by nature, and that was the reason why this histocompatibility arose. I don't think so. It's just a matter of the mechanism by which antigen is presented to a certain type of cell. So MHC is needed to present antigen to T cells, because T cells or the T cell receptor only sees antigen in processed form. And because you want to make sure that you see the most diverse array of antigens, you have to elaborate this peptide presentation system and make it structurally diverse to enhance the chances of each and every pathogenic peptide to be presented. And this is why perhaps there was pressure to diversify this presentation system. I'll come to this a little later in the series. So then it's basically an impractical outcome now when we do organ transplantation, that unfortunately the system is so efficient with this multi-allelic diversity of MHC, for example, that it is almost impossible to find somebody that perfectly matches the MHC in structure from one individual to the next. So I think that is not a mechanistic part of the system, it's just an unfortunate byproduct from a medical point of view, as it were.
You also told that thymus appeared during the evolution as a place for quality control,
Yes.
to eliminate autoreactive lymphocytes. Then how can you think about this idea when you remember about the ontogenesis of the thymus? It is large in small infants, and then it became smaller and smaller. So how does it mean that it is not necessary to eliminate autoreactive cells in adults?
No, no. The simple answer to this is again an efficiency problem. Once you have filled up your periphery with your long-lived T cells, and T cells are very long-lived, and if you are not coming down with infections, you don't have a reason to eliminate or to replace your T cells, because there are no cytopathic viruses, for example, that would kill your T cells. So you fill up your periphery, you don't have to produce them anymore. That's during infancy. You fill up the periphery is full, and then you shut down the production line. But the thymus is never completely gone, even in very old people. So if there is a need, and if you have a depletion of peripheral T cells, then the thymus comes back into function. So it's simply the production is reduced, just saving energy, because there is not enough trophic factors in the periphery to maintain larger number of lymphocytes, because they're always maintained at roughly the same level. And if there is no cytopathic infection or other use, as it were, or depletion of the T cell pool, then there is no need to make more. Yes?
Excuse me. Could you just tell to me what the number of lymphocytes in human organism? I just don't really know about what the number.
You ask critical questions. The trouble is we do not find volunteers. I don't have to continue that sentence, no? So we can make guesses from mice, for example, and then simply extrapolate to the bigger bodily volume of humans, or the blue whale, for example. But it's a little difficult to say, because even for one mouse, it's very difficult to process a mouse such that you get hold of all lymphocytes. The ones in the blood, of course, is easy. The ones in the lymph nodes and in the spleen and maybe in the thymus, that's all very easy. But the ones that are already in the intestine, it's very difficult to retrieve all cells. All the cells that are traveling the body are sitting just in my right quadriceps, for example. They are very difficult to get out.
Okay, maybe just in
rough numbers, you mean?
Blood.
Well, that you know. I mean, you can calculate that the blood is simple. It's 5,000 lymphocytes per microliter. That's the ballpark. But of course, then your next question will be how many T cells, how many B cells? Okay, good.
But I've got another question, just to clean my information about differentiation in thymus. Because does information, as could I understand that it has been eliminated about half of lymphocytes which produced in thymus, which come to thymus to selection. Yeah.
During this elaboration that I gave today, you mean? 50. Yeah. Yes. Mm-hmm.
Yeah. And is this information have evidence? Histological evidence when you just cut thymus in microtome and look at the number of apoptotic cells.
So, can anybody else answer that question? It is a paradoxical result. You do not see much evidence of apoptotic cells in the thymus. However, the thymus is full of macrophages, and macrophages can eat dying cells amazingly quickly. Okay? If you introduce a mutation in the gene that encodes DNase 1, that is important to digest nuclear DNA, so the macrophages need this because they have to eat, as it were, dying cells. Then they are, of course, filling up and they cannot do this, and then all of a sudden you see all these corpses of dying cells. And in fact, estimates go as far as 95 to 99%, depending on which source you consult, of cells that enter the production line. 99% are lost on the way and only 1% makes it to the end. So if you were the chief engineer of General Motors and you would present that to your board, this is my procedure. 100 cars I start with and only one I can sell at the end. You would not stay for a long time in that company. Unless you convince the board this is the most efficient, there's a big advantage for doing so. Because some rich customers are really keen on getting the perfect car.
So there were experiments when the macrophages
was incapacitated in eating these dying lymphocytes, yes, and then all of a sudden you see this.
Maybe it was an experiment when macrophages was, I don't know, like switching off the macrophage activity, and after that you can see a lot of apoptosis.
Yes.
Yeah?
Yes.
Oh.
Mm-hmm.
But this 99% is not just because of the autoreactivity, it's also some other functions.
Yes, there are some other problems, of course, as you know, when you start building a cupboard for yourself. I mean, you have to try several times before you get it done, and that's the same is true also for lymphocyte development. There are many steps that can go wrong. You might easily produce a non-functional receptor that is not at all functional, not even autoreactive, it's just nothing, just junk. And that, of course, that lymphocyte also has bad luck. So there's a lot that can go wrong, apart from producing self-reactive receptors.
Actually, a bit of a philosophical question maybe. So if roughly half of the lymphocyte develop cross-reactive immunoglobulins, right? Does it mean that the half of the potential epitopes are, self-epitopes, or because you would think that, the percentage of the antibodies for different epitopes are produced with the same, random chance. So if half of them are self-reactive, does it mean that half of the epitopes are self-epitopes? Meaning we have as much targets in ourselves as targets around us, like?
Yeah, if we ignore his argument that there is specificity of one receptor for more than one antigen.
Okay. Yeah.
Or reactivity, say. So it's a complicated issue. I wouldn't put any numbers on your argument, but of course there is some truth in this. If the antigens that are produced in the thymus, the self-antigens, would cover the entire repertoire of specificities, not a single cell would survive. Right. That's clear.
So it's actually just a portion of the potential.
Yeah, so it is between 1 and 0, somewhere in the middle.
So actually, and speaking of the predators, you gave the number for the lamprey, what was 10 to the power 14.
Yeah.
What would be the same number in humans?
Well, for some types of receptors, for example, these gamma delta T cell receptors, the numbers go up to 10 to the power of 19.
Mm-hmm. 19.
19.
Okay.
I once tried to do some calculations for the blue whale, and I just could not imagine, only when I was doing the calculations, these animals are really big. You should go to a museum and have a look at them, right, and then think what their lymph nodes are like. Yeah. Jesus. Okay. I have to say no more. This is amazing how many cells they can produce and orchestrate. We'll come to this a bit later, because the orchestration also is very important. Bring the right specificity to the right place.